Good afternoon.
Hello, yes, hi everybody. I'm just trying to get my picture up again. I cannot work out why this happens. Oh, do, do, do. Why won't it give me my video? Oh, for goodness sake. Man. It says I never have not. Oh, for goodness sake, no. No. Does anybody have any suggestions as to how I get my video up? There it is. But now, how do I do you all at the same time?
To be honest, I'm not even 100% sure.
Yeah, God, honestly. This always happens. Any case, I'm just going to try for 1 minute more, and then I'm going to just start and we'll just have to do without me. Can you see me?
Yes.
Yeah, we can.
Okay. Okay. As long as you, there I am. There I am. At least I can do it that way. It's difficult to talk when you can't see yourself. You feel like you. Okay, everybody. Hi, everyone, and welcome back. This is our last class dealing. This is the class will probably carry on till the next class, but this is now ending our discussion about metabolism and especially about energy and matter and how the cell manages it. Before we move on to discussing photosynthesis, which is what we're going to mostly discuss today, I just want to finish off our discussion about cellular respiration, because there's one aspect of it which we need to understand, and that is, we've talked about, we looked at that respiratory chain. We saw how the respiratory chain produces masses of ATP very efficiently because it uses electrons from the electron donors like NADH and FADH2. But what we need to understand is that that whole electron transport chain is operating because at the end of it is a very, very powerfully electron negative atom, which is oxygen. and it's effectively pulling all of those electrons all the way through that transport chain. And along the way, the electrons lose potential energy. They donate that energy to systems which pump the protons. So that system works. The whole efficient cellular respiration system works because we have oxygen at the end. And finally, those electrons and protons unite with the oxygen to produce water, which is why we breathe out water vapor. What happens if we don't have oxygen there? If you don't have oxygen at the end there as a terminal electron acceptor, that whole system stops. And the whole system stops all the way up to glycolysis. Glycolysis can, in fact, continue to work. It can continue to work, and it produces very little ATP, but it does produce some. It produces 2 molecules of ATP. For some organisms, that is enough. For example, in many of the prokaryotes can survive without oxygen. They can operate with oxygen or without oxygen. We say they are facultative anaerobes. So they can survive in the absence of oxygen just by using glycolysis. But there are many other systems as well, which can at least temporarily also use glycolysis to just provide just enough ATP for the system to keep, for the cells to keep going. In fact, even in our own cells, there are times when we resort to glycolysis. For instance, in our muscles, there are times when our muscles resort to glycolysis as the only source of ATP, temporarily. That's very short-lived. So almost all cellular respiration depends on the presence of oxygen. Without oxygen, most of it stops, but glycolysis will continue for a while. But there's a big problem. There's a big problem with using glycolysis as the sole source of ATP. So here's what glycolysis can do. Okay, there's the glycolysis system. It can produce 2 molecules of ATP. Remember, we have to put in some at the beginning to get it going. We have to put in 2. Do we make 4 altogether? So we have a net gain of 2 ATP. For some organisms, that is enough for them, like the prokaryotes. And in some cellular systems, that's enough temporarily for the cell to carry on. But here's the problem with using that. Have a look here. This is how we actually produce that ATP by glycolysis. We actually reduce NAD plus there. We reduce NAD plus to NADH plus protons, and that gets sent off to that electron transport chain for oxidative phosphorylation. Under normal circumstances, the cell uses up the NADH and restores NAD+. But if there's no oxygen, then the whole system stops right there, except for glycolysis. Glycolysis carries on, produces a little bit of ATP, but the problem is it runs out of NAD+. It converts all the NAD+ to NADH, and glycolysis has to stop. That's a big problem, but there's a way around it. And various organisms have found ways to deal with this problem. The cell needs to regenerate NAD+. In the absence of oxidative phosphorylation, there's very few ways to do that. But the cells have devised various strategies to do it. And this is what we refer to as the system of fermentation. Fermentation is nothing more than the cell's ability to regenerate NAD+ from NADH. And this is the way, are you all right with this so far?
Professor, I have a question.
Yes, go ahead.
So the first question I have is, this happens in the body, right? In the cell?
Correct. Yes.
And then also, like knowing what fermentation is there like an alcohol byproduct that's
We'll get there right now. We're going
okay.
to get there right now. Yeah. Yeah.
And then how does the body deal with it? Because I know like excess levels of alcohol can be dangerous, right?
Yes, yes, but it's not. Yeah, that's a different topic. Dealing with the alcohol is a detoxification problem
Okay.
that our livers do. There is a problem. I'll tell you now that there is a problem inside our own cells, which I'll describe in a sec. Okay, so here's the problem. Glycolysis, our only source of ATP in the absence of oxygen, but it uses up all the NAD+. We've got to somehow regenerate NAD+ from NADH. So the products of glycolysis is pyruvic acid. And what cells do when they ferment is they take this product, the pyruvate, and they change it to some other kind of product. They use up NADH in order to do that. So let's follow this one, pyruvate. Don't worry about the name. It makes this molecule called acetaldehyde. But the cell then takes acetaldehyde and converts it to ethanol. In the process, it releases carbon dioxide. Well, what organism, if you put it under anaerobic conditions, produces lots of carbon dioxide and produces ethanol? The answer is a yeast. This is how we generate alcohol from using yeast to do the fermentation for us. What the yeast is doing, they are regenerating their NAD+ by taking the products of glycolysis and changing it to ethanol. And what they're doing is they're reducing the pyruvate to ethanol. In the process, they release carbon dioxide, and they give us, that's two useful products for us from fermentation by yeast. Carbon dioxide, that's what makes your bread rise. Ethanol, that's what makes you feel happy on a Friday evening. Okay, so these two products are produced from fermentation by yeast. There are other fermentation systems. Many bacteria, for example, will produce a substance called lactic acid. And that is doing exactly the same thing. It's just that the pathway is a little bit different. They produce lactic acid from their pyruvate. In our own muscles, we can temporarily resort to a system very like this, where we produce lactic acid. And lactic acid in our muscles, when it starts to accumulate, that is what gives you the burn in your muscles. That is the signal that your lactic acid levels are building in the muscles. So that's exactly what fermentation is. Remember, whenever you're asked, what is the purpose of fermentation? To the cell, the purpose is to regenerate NAD+. Why did it regenerate NAD+? So that glycolysis can continue. Why do you need glycolysis to continue? Because you have no oxygen and you have to have a source of ATP. In these systems, this is an anaerobic system, a system that works without oxygen. These systems, the metabolism is usually slower, much, much slower under anaerobic conditions. For this reason, you're only producing very little ATP compared to the 30, 32 that you can produce from oxidative phosphorylation. Okay, I don't need, the book will discusses in detail the different kinds of fermentation. All I need you to remember is we can produce the different fermentation systems, produce various products. The yeast produces ethanol and carbon dioxide. Other systems may produce lactic acid, which in itself is very useful. Lactic acid is what makes cheese products. It's what makes things like sauerkraut, all of those fermented foods very often use a lactic acid producer to do it. So that's the one solution when there is no oxygen, is to resort to fermentation. But there is another solution as well, which is also quite common in the prokaryotes. And it has a strange name. It's called anaerobic respiration, which sounds like a tautology. How can you be respiring under anaerobic conditions? And the answer is that they are not using free oxygen the way an aerobic system does. Our own cells, for example, we depend on free molecules of oxygen to drive that oxidative phosphorylation. But many prokaryotes are able to use another an ion that is rich in oxygen, such as sulfate or nitrate or nitrite. And they use that as the terminal electron acceptor. What they're really doing is they're using the oxygen within that ion as the terminal electron acceptor. And it works very well. It's not as efficient as aerobic, completely aerobic respiration, but it's much more efficient than fermentation. So just remember, in the absence of oxygen, cells can still resort to different strategies to try and continue to produce ATP. One is just depend on glycolysis and in fermentation. The other is resort to anaerobic respiration. Okay, and I think that the very last thing I want to say about cellular respiration, your book will go into considerable detail about this. I'm not going to discuss it in major detail. I just want to point out two things about this whole system, all the way from glycolysis all the way through to oxidative phosphorylation. There are many, many, many different pathways in the system. There are many metabolic pathways. And that presents us with an opportunity to use different substances as fuels, because with slight modifications, we can slot these in two pathways as a substrate. So, for example, if we take fats and lipids, slightly just modify them, extract the fatty acids and modify them very slightly, and you can start pumping them into different pathways in this whole system. The same carbohydrates we understand really well, because that is what we've discussed for glucose. You can even use proteins as a source of fuel, and put them into different parts of the pathway as well. I'll point out to you, the carbohydrates are the most easily used, because, for example, in us, we've discussed the use of glucose, but storage products in plant starches, in animals is glycogen, and those are carbohydrates. They're simply polymers of glucose. So that's our most readily accessible, our easiest, but also our most rapidly used resource for fuel. Our long-term storage, we depend on fats. Those fats are usually laid down specifically to be extremely stable and only used essentially under starvation conditions. If you are starved, the first thing you use are carbohydrates. The next thing are the fats and lipids. But then if you are extremely starved, you go into a very dangerous place where you begin to use up proteins, and in particular proteins of muscle first, and ultimately functional proteins like proteins in nerves and those sort of things. And you can end up with really severe damage as a result of that degree of starvation.
Professor?
Yes, go ahead.
Let's say if you don't eat for a certain time, I know glycogen is stored in the liver to produce,
correct,
right? This can also be used, right, for cellular respiration?
Absolutely, absolutely, yes. That's your first resort, is to mobilize glycogen. That you'll do the whole time. You'll mobilize glycogen stocks, and they'll just go straight into glycolysis as simple sugars.
And then after you eat, you restore that glycogen, right? That's stored.
Correct. Yes. When we eventually talk about the endocrine system, and we'll learn about the importance of insulin in regulating blood sugar. And that is what one of the responsibilities of insulin is, to ensure that we keep a level of glucose in the blood, of sugars in the blood, which we can use, and that the rest is put away as glycogen and stored in cells.
Wait, Professor, I have a question.
Yeah.
I'm sorry.
Yes, go ahead. No, hi.
So long story short, what you're saying is, like, as like you start to like basically starve, like you got to start burning off different things. First it's the carbs, and then you got to burn into the fats, and then eventually it can get bad to go into proteins, is what
Correct.
you're saying?
Yes,
All
that
right.
is correct.
All right, thank you.
Mhm. Okay, so now we need to also talk about where on earth our organic molecules come from, because we are heterotrophs. We have no way of feeding ourselves from simple things like carbon dioxide and water. We depend absolutely on organic molecules, which already contain the atoms that we need to build our biomolecules, but which also contain the energy that we need to drive our metabolism for all of our metabolic processes. And all of that originates in this incredible process called photosynthesis, which is the responsibility primarily of plants on the planet. So it behooves us to understand something about photosynthesis and to understand its relevance to the biosphere. The first thing to remember is that in the course of metabolism of anything, any living thing, metabolic processes are inefficient. And as a result, living things continually waste energy. It's a requirement of the second law of thermodynamics that, unfortunately, every energy transformation, every energy transfer is inefficient. And some of the energy is lost as heat that is ultimately radiated out to space. So the upshot of that is that energy flow on our planet is one way, in and out. We have to continually replace the energy that is lost through the inefficiency of metabolic processes. If it were not for the processes that performed by things like the plants, we would run out of energy. But fortunately, this group of organisms called the autotrophs are able to take inorganic molecules, carbon dioxide and water, and they are able to take energy, in the case of photosynthesis, the sun's light, and they are able to convert sunlight into chemical energy, potential energy, in the form of bonds, in the form of chemical bonds. What they're doing is they're taking a simple thing, carbon dioxide, and they are reducing it. They are extensively reducing it. The simplest way to think about it is that they are reducing carbon dioxide with hydrogen to produce sugars. And it is on those molecules that everything else depends, ultimately. So plants are continually replacing the energy which is lost through the inefficiency. And at the same time, they are taking available molecules, carbon dioxide, water, and other atoms as well, ultimately, building them into biological molecules, which everything else can then depend on. We can eat plants, or we can eat things that have already eaten plants. But what we're actually doing is we're accessing the molecules and the energy which plants have put into those biological molecules. So these processes of autotrophy, self-feeding, like photosynthesis, actually feed all of us. I say these processes, we are going to talk only about photosynthesis because photosynthesis is the most important. The bulk of autotrophy on the planet is done by photosynthesis. There are some other forms of autotrophy. Things can use chemicals as a source of energy and then take carbon dioxide and water and put them together. but we won't talk about that. Is chemoautotrophy, and we're not going to discuss it. It's a minor part of the budget for the planet. Photosynthesis is by far the most important process of autotrophy. So that word autotrophy, it just means self-feeding, self-sustaining. And remember that what they're doing, they're taking inorganic, simple inorganic molecules, carbon dioxide, water, and they're putting them together to form complex organic molecules with lots of energy locked into the bonds. So if we look at what does this, the first thing is the vast bulk of photosynthesis is performed using a system called plant photosynthesis. That's its name. The actual metabolic system is plant photosynthesis. I'll show you why we distinguish in a minute. But obviously the plant, all of the plants, all of the algae, both marine, freshwater, many single-celled organisms, protists, plankton, and everything in the sea. These are our most important photosynthesizers, and they all use this system called plant photosynthesis, which originated in this group here, the cyanobacteria. So the cyanobacteria are they're bacteria, but they have a complete photosynthetic system. It evolved in them, and it was handed over in evolutionary time. It was handed over to everything else, and it evolved as such an efficient system that really there's not a lot of difference between this kind of photosynthesis that bacteria perform, cyanobacteria rather, and that performed in the higher plants. I should just mention that this is a reflection of the fact that in the cells of these organisms, there are special organelles to perform photosynthesis that might be derived from bacteria. And we'll mention that again in a minute. So this is plant photosynthesis. I just would mention there are some other obscure kinds of photosynthesis in the prokaryotes. Purple sulfur bacteria, you don't need to remember this, but just remember the most of the photosynthesis is plant photosynthesis, but there are one or two other kinds of photosynthesis that have evolved. So we'll confine our discussion to the plants and mainly to the land plants. And first of all, realize that when we look around us, the first thing you notice is they're all green. Green is the common kind of feature of organisms which are performing plant photosynthesis, and for a very good reason. And that is that plant photosynthesis depends absolutely on a molecule called chlorophyll. There are different kinds of chlorophyll, but the chlorophylls are all basically green in color. And that's why photosynthetic organisms are distinguished by that color. For most of our land plants, the organ of photosynthesis, the major organ of photosynthesis is a leaf. But remember that many other parts of a plant can also be photosynthetic. Stems can be. Some plants, like cacti, don't have leaves. It's only the stem that is photosynthetic. But it all works on basically the same kind of system. So if we examine a leaf, we look at its anatomy, there are certain features that tell us a lot about what is required for photosynthesis. Let's have a look. This is a leaf in section. Here's the top there. Here's the bottom here. Both called the epidermis. They're up top and bottom. And that's quite tightly sealed. It's basically gas-proof, excepting for the fact that it is penetrated by these pores called the stomata. Much more common on the underneath of the leaf than on the surface, although you do get some on the surface as well. These are controllable. They can be opened and shut in response to the needs of the plant. if you look on the inside, the first thing that strikes you when you look at a section of the leaf is there's a lot of space. And that's deliberate. It's so that gases can circulate easily inside the leaf. Because don't forget that photosynthesis needs carbon dioxide. So carbon dioxide has to get in. It also needs water, but water is absorbed through the roots and distributed through the plant and is evaporated as water vapor here inside these spaces, so that it's available to these cells as well for photosynthesis. So you've got carbon dioxide and water vapor circulating in here, but the very important product of photosynthesis is oxygen. And it's very important that the oxygen be vented from the leaf, because high levels of oxygen impede photosynthesis. So the oxygen has to percolate out and carbon dioxide has to percolate in. Together, they're going to be put together in the process of photosynthesis. And that process in the plant is, here's a whole cell from inside here. Here's a whole cell. And if you look inside, these little organelles there all over, each one is a chloroplast. And that is where the process is actually going to be performed. I think I've got a bigger, yeah. So the chloroplast is a very highly organized organelle. First of all, it has a double membrane around it. And inside the chloroplast here, you'll see that there are these stacks. They look like envelopes that are stacked on top of one another. Each layer is called a thylakoid. And they're piled up like this into a collection called a granum. So that whole thing there is a granum. It's a pile of thylakoids. And look where the coloration is. You'll see that it's actually this membrane here that is colored green. That is because there is masses of chlorophyll actually embedded in that membrane. And again, think back. If you understood all of the principles of the functioning of the membrane in cellular respiration, all of that electron shuttle and how the energy in the electron was harnessed to pump protons, all of those principles apply in photosynthesis as well. We're going to see some similar kinds of systems operating. This here is an electron micrograph of a chloroplast. And again, there's a granum, a pile of thylakoids all together. They're all connected. One or two other things about the chloroplast, though. Like a mitochondrion, the chloroplast has its own DNA as well. The chloroplast has its own ribosomes, and it produces some of its own proteins. Not all of them. It's dependent on the cell for quite a lot of things, but it can produce some of the things it itself needs. And its its chromosome looks like a bacterial chromosome. It's a circle, just like a bacterial chromosome, which makes us think that the chloroplasts in plants originated as free-living prokaryotes in evolutionary time, and were acquired by a primitive eukaryote cell to go on to become the chloroplast. That's that whole evolutionary system called endosymbiosis, where both mitochondria and chloroplasts, for example, are thought to have once been free-living prokaryotes. Okay, so this is our chloroplast. Yes, go ahead.
So you mean that before, like when the evolutionary process started, that these were they were their own organisms?
Yes, yes. There was a primitive eukaryote cell. There were primitive eukaryote cells that acquired either mitochondria on their own or mitochondria and chloroplasts. And those that acquired mitochondria and chloroplasts went on to be able to perform plant photosynthesis depending on the chloroplast for the photosynthesis. Okay, so yes, we're going to have to talk about the chemistry a little bit, but I'm keeping my whole discussion of photosynthesis relatively simple. I think that you got a very good idea about how these systems work by studying cellular respiration. And as I say, if you understood that, you'll understand a lot about photosynthesis, excepting for one thing, and that is in respiration, I'll just jump down here. In respiration, what we did was we took something like a sugar, and we took oxygen, and we processed it in such a way that we accessed all of the energy in these hydrogen bonds. And we ended up producing carbon dioxide and water, a kind of a waste product. But in the process, we were able to access the energy in these bonds, and we were able to produce useful biological energy. So that's the process of photosynthesis, of respiration. Now let's look at photosynthesis. Look at this. 6 CO2 plus 6 H2O plus light energy forms, this is glucose, and releases oxygen as a waste product. That is the reverse of that. This respiration energy, this respiration formula equation is actually the reverse of the photosynthesis equation. And that is almost exactly what happens in photosynthesis. Because here we are extracting the energy from these bonds, we're actually oxidizing our fuel. Here we are reducing these substances to produce a substance with lots of hydrogen attached to it. And in those hydrogen bonds, there is a lot of energy which is derived from light. It's the light. This is the magic of photosynthesis. It transforms that light energy into the chemical energy of these bonds here. Now, I will just tell you one thing. If you look at this formula in your book, you'll see something a little bit strange. They often put 12 H2O and then put another 6 H2O over this side. The only reason they do that is because this formula gives you the idea that all of these oxygens and hydrogens come from one source, but they don't. There's actually a complete reshuffling of the oxygens and hydrogens in this process, and it's a bit difficult to say where, it's not difficult to say, it's difficult to track where the hydrogens and oxygens come from. So they sometimes include another 6 water molecules to try and explain it. I have just used a simple equation here. This is all you need, remember, this simple equation here. And it fits nicely because it looks like the exact reverse of the respiration equation.
Professor?
Yes, go ahead.
The end result after C6H12O6 glucose being produced plus 6 oxygen molecules?
Correct.
Okay.
Correct. Oxygen is a byproduct. It's a byproduct of photosynthesis. And it actually is, there's an interesting story attached to the production of oxygen in evolutionary time. When the cyanobacteria evolved the process of photosynthesis and began to release masses of oxygen into the atmosphere, that is what actually changed the atmosphere completely. Up until that time, there had been no free oxygen. It was the process of photosynthesis that actually provided all of the oxygen in the atmosphere. But there was a problem with that, and that is oxygen is a very dangerous molecule. It's a very, very dangerous molecule to have around, especially around biological molecules. It tends to just produce very, very reactive species that break up biological molecules. And the system here of respiration may well have evolved to deal with the oxygen, to deal with the dangers of oxygen. It was only it was a fortunate aspect of that you could extract energy from it. But it was actually an attempt to escape the effects of oxygen. That's the story. That's the evolutionary
So the
story.
byproduct, oxygen gets released into the atmosphere, but the glucose, is it utilized by the plant?
No, well, it's how you mean in the plant itself? Very
Yeah,
much so.
I mean,
Very much so.
they use it for
Yes.
energy, right?
Yes. Yes, that's a really important point. People forget that plants must also be respiring. They have mitochondria to produce ATP. So this system of photosynthesis is making these energy-rich, atom-rich molecules, but the plant still has to use them. The plant must still use them for its own metabolism. And it's easy to forget that plants must be respiring at the same time as they are photosynthesizing. They produce much more than they need, so, under normal circumstances. Okay, so remember this, that that the system of respiration was exergonic in the end. We ended up extracting energy from it. It's a catabolic process. Photosynthesis is very much an endergonic process. It uses actually a tremendous amount of energy. It needs a lot of energy to make those molecules. But who cares? If you're a plant, the energy is free. It's sunlight. As long as the sun is shining, you have access to a source of energy. So it doesn't matter how expensive energetically it is, as long as you have that constant source of energy constituted by the sun. So when we look at the, yes, go ahead.
What happens if there's no sunlight? Like if it's a rainy day, are they still using the natural light?
They will use whatever is available to them. But for example, in the dark, photosynthesis stops, but respiration carries right on. All the normal processes of the plant, of the organism, carry right on. And then when in the light again, they start photosynthesizing. And as I say, most plants produce a lot more than they use, and they store everything. They store a lot of what the extra as starch, for example, which they can access at any time. So we call the plants, that word producer is a real term. We actually say that the plants are the primary producers. They are the source of feeding for all of the biosphere. They are the primary producers. And if we look here, you can see why. You can see how it works. What they're doing here, they're taking carbon dioxide and they are reducing it. They're adding hydrogen, electrons and protons to it. And that is derived from water. And water is what becomes oxidized. And if you oxidize water, you remove hydrogens from it. And what is your byproduct? It's free oxygen. So this is the formula for photosynthesis. Sunlight energy is used to reduce carbon dioxide with hydrogen, and oxygen is the byproduct. And this is formally the product of photosynthesis, and this is glucose. I say formally because, as I'll describe in a while, there's a couple of steps to actually producing the glucose. Okay, so the process of photosynthesis is divided into two sets of reactions. And the first set of reactions are absolutely dependent on light. They will only work in the light. The second set of reactions do not need light. So the first set of reactions, what we're going to do actually is we will make a little bit of ATP, but we're going to make masses of reducing power. Now, I'll just point this out to you. In the plants, the electron carrier is this substance, nicotinamide adenine diphosphate, and it's NADP+. It's very much like NAD, excepting it's got that phosphate group attached to it. NAD nicotinamide adenine dinucleotide with a phosphate attached to it. But it works exactly like NAD plus in respiration. It's just a feature of the plants that they use that. Okay, so our light reactions are all performed in the thylakoids of the chloroplast. The light reactions are actually fixing energy, and they're fixing a lot of energy, because what is going to happen next, here they're going to make ATP and NADPH. What happens next is that we have an entirely separate set of reactions. These are not dependent on light. You may sometimes see them referred to as the dark reactions, but there's no requirement that they be performed in the dark. It's simply that they don't need light. This is a very complex cycle. We will only discuss it in broadest detail, broadest outline. But it's called the Calvin cycle, named for the person who described it. And it fixes carbon dioxide. It takes free carbon dioxide gas, and it will reduce it. And it reduces it to a set of products which ultimately end up producing a sugar. So we've got these two sets of reactions. Light reactions, what they're doing is their main responsibility, take that to sunlight energy and put it into chemical bonds of some sort. Then use all of this energy to fix carbon dioxide and to create this complex product. It's carbon dioxide, which has been extensively reduced to form a sugar. But that is very, very, very expensive. It takes an awful lot of energy, as you'll see in a while, just to produce the molecule of sugar. But it doesn't matter. It absolutely doesn't matter, because this energy is free. It doesn't cost the plant anything. It can perform all of these very energetically expensive reactions because it has a free source of energy, as long as the system is working properly. Let's just have a discussion a little bit about chlorophyll and about pigments in general. There's a couple of things which are sort of counterintuitive when we talk about these pigments. First of all, we have to ask the question, why are plants green? Why is chlorophyll green? Well, a pigment is any substance which interacts with specific wavelengths of light. If you're painting and you paint a beautiful blue color onto a piece of paper, the blue is blue because it is reflecting blue light. It's absorbing all the other wavelengths of light, but it's reflecting blue. Same with chlorophyll. Chlorophyll is green because it reflects green light. Green light is reflected from it. The other wavelengths of light interact in another way with the pigment, with this pigment chlorophyll. And first of all, let me just tell you, chlorophyll is not the only pigment in the chloroplast. There are a whole lot of different pigments. Some of them are there to absorb excess light, effectively to prevent burning. But there are several pigments which are there specifically to absorb different wavelengths of light that chlorophyll cannot. And they absorb those wavelengths of light and contribute that energy to chlorophyll. It's a complicated kind of system, so I'm not going to discuss it in any more detail than that. I'll just show you here. This is what we call an absorption spectrum. This is, we're going to measure on a spectrophotometer what wavelengths of light are absorbed by the different pigments. We'll extract all pigments that we can from a plant, separate them, have them in solution, and then we'll pass different wavelengths of light through them and see what is absorbed best. This is one big group of pigments called the carotenoids, and you've probably heard of them. The carotenoids are yellow, orange, color because that's the color that they reflect. But here you'll see this is these are the wavelengths of light. We're watching how much of this light they absorb, not how much they reflect to give them their color, but how much do they absorb. Well, the carotenoids, have a look here, they're absorbing best around about here. And that is the blue into violet color of light. Here's one species of chlorophyll, chlorophyll A. Have a look here, and you'll see it's absorbing best down here in the violet. This is chlorophyll B, another species of chlorophyll, and it absorbs more in the blue. But carry on following the chlorophylls, and you'll see they don't absorb here. Why? Because this is green. It's the green and yellow, which is actually reflected. We see it as green. Okay, this is reflected from chlorophyll, but it does absorb down here in the red. So the chlorophylls absorb here, blue, violets, and then down here at the reds, and everything else is reflected. The carotenoids, they absorb all around here, and they will actually also contribute to the chlorophyll. They will contribute energy to the chlorophyll. You'll see the reason for that in a second.
Wait, Professor, I have a question.
Yes, go ahead.
So, according to the first chart that you're showing us right now, is that like the highest peak of each of the graphs? Is that like where it's like absorbs the most?
Correct. Yes, that's its best. That's its best absorption.
So, if like for chlorophyll, it would be like a purple bluish.
Correct. Yes, and it depends on which chlorophyll you're talking about. One of them is much more down into the violet.
Oh,
chlorophyll A is down to the violet, and chlorophyll B is in the blue.
Okay.
Yeah. So there's another way to think about this, and that is to actually shine light of different wavelengths onto a plant and see where it produces the most oxygen. That's a measure of its rate of photosynthesis, right? So let's do that experiment. We're going to shine light of varying wavelengths onto a plant, the same plant. We're going to observe where does it perform photosynthesis best. And unsurprisingly, look here, it has a peak here, which corresponds to all of these. It corresponds to the best absorption of these pigments there. And look here, it has a peak here corresponding to this absorption here. It looks like the absorption of the light by the pigments is what is driving the photosynthesis. If it's absorbing best down here, it's doing the most photosynthesis there. If it's absorbing best here, it's doing most photosynthesis there. There's a dip here because this is light which is reflected. It's not absorbed. So there's no minimal photosynthesis carrying on down here. I'll just tell you about this little experiment. Just because it was kind of clever. This is a very, very old experiment, which was done by somebody who was studying photosynthesis. That's the called the action spectrum. It's the result of shining light of different wavelengths onto the plant and seeing how well it photosynthesizes. Well, this Engelmann was a worker he was a botanist who was studying photosynthesis, and he came upon this easy little experiment. And so what he did was he took an ordinary prism, which creates a rainbow, and microscopically he projected that rainbow, and he put a filament of alga in it. This is an alga called Spirogyra, which you will see when you do your biology labs, you'll see Spirogyra. So Spirogyra is photosynthesizing, but the filament of alga is lying in different wavelengths of light. Okay, and then he the clever trick was he put aerobic bacteria in. Aerobic bacteria have a requirement for oxygen, So they're going to cluster where the oxygen is highest. And look at this. This is how the bacteria clustered, there and there. Compare the bacterial clustering to this action spectrum here. Isn't that neat?
Yeah.
So this is a real reflection of the fact that down here, the plant is producing much more oxygen than it is producing here. Much more oxygen here than it's producing there. It's a clever little experiment. Okay, so we need to just talk a bit about the nature of pigments, and especially about the nature of this master pigment of chlorophyll. As I've told you, the chlorophyll is, we observe it as green because it reflects green light, but it absorbs widely, as we've seen, blue, violet, and then red as well. But we need to ask what is special about this absorption? What is it doing? Well, the trick is that the chlorophyll molecule has electrons associated with it, which can be boosted to a higher energy level. They actually move to a new orbital pattern when light shines on them. The light is absorbed by the chlorophyll, and one of or more of its electrons are boosted to a higher energy level. Now, if we simply leave, let's say we get a solution of chlorophyll just by itself, and we shine light on it for a good length of time, and then we switch the light off. What will happen is the process isn't instantaneous. It goes on for a little while. When we shine light on it, the electrons are boosted. Switch the light off, and the electrons begin to fall back down to their ground state, to their normal energy levels. They've gone there because they had energy input. They come down, they release that energy. And they release it as a small amount of heat and as a small amount of light. And we can see that. Here is a solution of chlorophyll. It's had light shone on it for a certain length of time, and then the light's been switched off. And it releases light. It is fluorescing. It's actually glowing. It's glowing red because the electrons in the chlorophyll are falling back down to their ground state and releasing energy.
Wait, so Professor, I have a question.
Go ahead.
So you're saying with the chlorophyll, what happens is when the light is shined on, well, if you use like a light and shine it on chlorophyll for a good amount of time, and then you shut it off, it's going to go from green to red because the...
No, not quite, not quite. It's actually, it glows faintly in the dark. That's what you're seeing here. It's actually glowing red. If you were to shine light back on it, it's still green. But it actually is releasing, you've boosted electrons, and when you take away the light, they all fall back down to their ground state, and they release the energy as light and a little bit of heat. And that's what you're observing here. So now, what you need to think of, wouldn't it be useful if we could harness this energy? If we can devise a system where we shine light on chlorophyll, excite its electrons, let them fall back to the ground state, but control that fall and harness the energy that is released. Well, that is exactly what happens. All of the systems of photosynthesis, this is what they depend on. They depend on the fact that chlorophyll molecules have electrons which can be boosted and then can be allowed to fall back down to their lower energy level, and the cell will harvest that energy and use it. And that's what we're going to watch happening.
Okay.
Okay. So when you go to your textbook, you'll see there's an awful lot of extra information that is associated with what I'm going to tell you. I'm keeping the story relatively simple. I'll just do one run through now, and then we'll stop so that you can ask me questions for your test, etc. But we'll just do one quick run through, and don't panic, because this is not on your test this time, and we will repeat this at the next class, so to make sure everybody has it clear in their minds. So the chlorophyll and the pigments in the chloroplast are very, very carefully arranged. They are carefully arranged in the membranes of the thylakoids inside the chloroplast. They're organized so that groups of pigments lie really close to one another. Now, I'm not going to go into the details. It's called, they form what is called an antenna. And it's a really useful name because you can picture it like a radio antenna that is actually gathering light. Instead of gathering radio waves, the radio antenna is gathering light. And it's actually focusing that light in a way, not quite, but in a way. Because have a look here. There are two light gathering complexes. They're called PS2 and PS1. The names are unfortunate. It's just when they were discovered. PS1 was discovered before PS2, but we actually talk about PS2 first. It's the first one in the scheme. That's a little bit unfortunate. Okay, what I want you to have a look at, first of all, here in the center of this light gathering complex, there is a molecule of chlorophyll. It's a species of chlorophyll called P680, which is its favored wavelength. So there it is, sits there, and it has electrons which can be boosted. They can be boosted directly by light shining on them, but they can also be boosted by light which is shone on all sorts of other pigments that surround them. Those pigments actually contribute all their energy to P680. They do it in a complicated way. It's called quantum harmonics. I'll just explain it like this. You can picture it like, here's a pigment molecule. Light shines on it. It begins humming with energy. That humming causes these all to start humming. And ultimately, the hum is handed over to this molecule. So we've got lots of pigment molecules all gathering light, all contributing energy to this master chlorophyll molecule here. When it reaches a critical point, one of its electrons is boosted to such a high degree that it can actually reduce this substance here, which is the primary acceptor. Never mind about its actual name. Just remember the primary acceptor. This electron is accepted. It reduces this primary acceptor. Realize that this molecule here, P680, has been oxidized. It has lost an electron. It becomes P680 plus. It's effectively lost a negative charge. So it's now P680 plus, and its electron is here. That electron, any electrons that are lost by P680 need to be replaced. And they are immediately replaced by the fact that the plant is able to split water. There's a molecule of water there. And the plant is able to enzymatically split water and get the their electrons from water, and they immediately are contributed back to P680. Now this here is this really important step, because look what happens when you oxidize water. We're oxidizing it because we're taking hydrogens away, right? We're taking electrons away from the water. Our byproduct is oxygen. That's why oxygen is the byproduct of photosynthesis. That is where it comes from. And I should just mention, this whole system here is very poorly understood. Normally, to split water like this, oh, it's incredibly difficult. You have to use masses of electricity and all sorts of things if we try and do it industrially. We'd love to know exactly how to do this. It would be a great source of energy. But the plant does it. The plant splits water here to gather electrons to donate back here. And immediately they're back here, of course, they can be boosted again. Those electrons can be boosted. So now this primary acceptor has been reduced by electrons. This should be familiar to you, because what happens to this high energy electron now is it tumbles down through an electron transport chain in the membrane. It tumbles down that electron transport chain, and as it goes, it loses energy, which is harvested. And the energy is harvested for exactly the same purpose that we saw before in cellular respiration. In this case, it's to make ATP. So we hear light shone here, electron was boosted, tumbled down the electron transport chain, and it's used to produce ATP. That electron story is not finished yet, because we have another light gathering system. It's called photosystem 1, and it has another chlorophyll molecule. In this case, it's called P700. Again, its favored wavelength of light. P700 works exactly the same way. The pigments around it contribute energy, and its electron is boosted to a high energy level to another primary acceptor. In this case, P700's electron is replaced by this electron tumbling down here. This electron tumbles down here and reduces the oxidized P700. The electron from P700 now has entered another transport chain. And what happens now is that instead of producing ATP, excuse me, I'm just moving your faces out of the way. I have them in the wrong place. Instead of producing ATP, the second chain produces the substance NADPH. NADPH, well, that looks just like NADH, right? It's exactly the same. It's reducing power. So when we look at this whole system, this whole light system, we watch these electrons as they tumble through, and we see that what they're doing is they're forming high energy bonds, high energy compounds, which can now be used as a source of energy in the next set of reactions. I will just point out, I just thought of something that I needed to point out now. Oh yes, I want you to observe that we're going to be following protons as well. NAD, just like NAD+, when NAD+ was reduced, it produced NADH and a proton. So same with NADP+. When it is reduced, it produces NADPH plus protons. So this is a simple analogy for you, this diagram. We can have a look at a photon. I mean, at an electron. Its energy is boosted by sunlight to a very high level. It tumbles down through an electron transport chain. The first electron transport chain makes ATP. That electron actually replaces another electron which has been boosted to a high level here, and that electron is used to make NADPH. And with that, I think we can stop here because this sets us up for then discussing the other reactions, the reactions of fixing carbon dioxide, which are going to ultimately make a sugar, and we'll discuss that next time. For the moment, your brains are probably full anyway. But if anybody has questions, especially relating to stuff that will be coming up in the test, feel free to go ahead. Anyone? Edward? Edward always has such good questions. Edward, Nicholas, you always have such good questions.
Sorry, I was just writing down notes. I was conscious.
No, that's fine. That's fine. That's fine. I wasn't yelling at you. I promise.
No, I'm here. I'm writing down notes too.
Okay, that's good. I'm glad. No, I don't mind if people don't have questions. I don't mind at all. I just, for the moment, I really feel that I don't want to proceed to discussing the rest of photosynthesis, because I think that I honestly think you've probably had enough to absorb for the day. I will tell you that the little bit about fermentation and anaerobic respiration is included on the test. It is included with test material you need to study. Photosynthesis is not. So.
Well, I have a question.
Go ahead.
Would it be possible if you, for like the next 10 minutes or so, just go over exactly what's going to be on the exam on Wednesday? Like, not everything, but like just like the general ideas, like what we should know.
I don't quite know how to do that. Hold on 1 second.
Yeah, no problem.
Let me just think.
Professor, enzymes will be on the test, right?
I beg your pardon?
Enzymes.
Yes, it's membranes, cellular respiration, and enzymes, yes. It's membranes, metabolism, and then cellular respiration. Those are the 3 chapters.
So it's on 7, 8, and 9.
Correct.
Okay.
Yeah.
So just review the PowerPoints and just like look over our notes that we have in our book and.
Yes, and go to the assignments. Go to the assignments.
And the assignments. That's actually just what I'm about to say.
Use the assignments as the basis for your revision. If you go through the assignments, you do all of those exercises, that will take you right through everything you need to know.
Okay.
And then if you're sitting there with the PowerPoints next to you, if there's anything that you don't understand, find that video and just listen to me again explaining anything that you didn't understand. The more you repeat the stuff, the more I'm sure you've discovered that already. Your first hearing of things, you can be confused. The second hearing, oh, wait a minute, yes, I'm making my way. Third hearing, oh, yes, I understand. That's just how our brains work.
No, yeah. All right, that makes sense. All right, thank you.
Okay. I wanted to just try and find, where am I? Oh, I can't find it. Anyone else? Okay, so I will just tell. Yes, go ahead.
We need to remember that all enzymes are proteins, but not all proteins are enzymes, right?
Correct. And in fact, not all enzymes are proteins, because RNA, which is a nucleic acid, can also function as an enzyme. But really 99.9% of our enzymes are proteins. RNA is the exception. It functions as an enzyme, but it's not a protein. That's the only one. Did that make sense? I hope so.
Oh, and also I saw on Canvas that you pushed not the Canvas, on our textbook thing, my mastering thing that we have. You pushed back the assignments to a later date.
several students asked me if I could extend the dates for them.
Okay.
And I'm quite happy to do it. Yeah, I think I've extended them all by a month.
Yeah, I saw that. Thank you.
And I don't have any beef with them coming in late. I did set shorter dates at the start to try and push people into actually getting used to doing them. Otherwise, what happens is people think they've got a long time, they delay doing it until, you know, it's of no value to them. I will just mention one thing. You have a bit of extra credit coming up, and it's going to be for 2%. It says here 10 points, but it's actually going to be for 2%. Don't worry about this until after your test. But it's just a comparison between the functioning of the mitochondrion and the functioning of the chloroplast. It's a simple comparison between the two. And again, that'll be for 2%. Extra credit, that just means it's added onto your, whatever your total is, it gets added onto that. In other words, pay, you know, as I'm discussing these, you know, pay careful attention, because it'll help you there. Okay, then, everybody, if there's no more questions if you come across stuff as you're revising in the next couple of days, then you can always email me. I'll be happy to answer by email. Otherwise, I wish you luck on Wednesday.
Wait, Professor, I have a quick question.
Yes, go ahead.
Are we gonna have a quiz this week?
No, you've got a test on Wednesday.
Oh yeah, that's what I meant, a test, because we didn't have one last week, so I was just double checking.
No, you've got a full test on Wednesday, not just a quiz.
Okay, thank you.
You have a full test. You should have got emails from me about that. Everybody, please check your email addresses, because it's virtually my only way of being able to contact you efficiently. Canvas is not an efficient way to contact you, it seems. All right, nothing else? Okay, see you all on Wednesday. Well, I won't see you, but I'll be there with you, believe it or not, while you're writing, okay? And contact me if you run into any problems. All right, thanks for being here.
Thank you, bye-bye.
Bye-bye.
Bye, Professor, take care.
You too. Stay
Thank
well.
you.